is that Hobart? All will jump in when they do need to. So thank you. Thank you all for being here. As I mentioned a couple times, the nature of our discussion is a community discussion. We're incredibly broad and rich and diverse, well-educated as a community. There you are, Hobart. Did I hear you?
Hi, how are you?
Hi.
Hi, everybody.
You can't get the pictures on
Yeah. Hobart, do you want to start us off, or do you want me to?
All right, absolutely. Hello, everybody. Welcome, and thank you again for joining us. We have a really wonderful evening planned. Of course, Dr. Hock, and also some other special guests. Just wanted to keep you abreast of current changes that are going on here in the community. Uhills.org has continued to update the webpage so that there's lots of new information about Zoom meetings, things to do in place of normal activities. There are also some exercise classes and yoga classes, things like that that are available. All that information is there. Also, postpartum and pregnancy groups are meeting, groups that help folks with dealing with anxiety and grief. Those are also meeting. So keep your eyes peeled for information for ways in which we can maintain our virtual community. A couple of things that this weekend, there's a wonderful concert that happens normally on Friday. Today it was on Saturday at O'Keefe in Murasaki. A classical music pianist performs there, and everyone stayed nice and safely apart. And if you check out the Facebook page, you'll see that the Easter Bunny came by. So that's something to check out. Also, there was a little impromptu social distancing wake for John Prine, which folks sang a couple of John Prine songs. So we're sort of hanging in there and staying together. And keep your eyes open. Check out uhills.org for information about events and ways in which we can stay connected. I'm going to turn it back over to Susan and this evening's program.
Thank you. Okay, so as I was saying, thank you, Hobart, for that. Those of you who may not know, Hobart is the chair of the HRB and my favorite leader. So as I was saying, we're an incredibly broad and rich and diverse, well-educated community. So we're here to give answers to questions that are on the foremost mind of residents. And I hope that that's what we'll be able to accomplish tonight. We have less than an hour now, so I'm going to get to it quickly. But before I introduce our speakers, I want to let you know that you will remain muted so that we can go through our list of questions. We can't make this a discussion in the way that we would like to, but we will be always taking your questions. We have quite a few questions tonight, and if there is additional time to take them in the chat, then Eric and Michaeli can help me with that. The questions in general reflect several categories. One is general safety and containment. The other is diagnosis and treatment, the statistics and spread, and COVID immune response. And Dr. Cindy has taught us that we know that balance is important now, so we will not go over 1 hour. So allow me to introduce our speakers. Dr. Cindy Hock is the chair of family medicine and champion for primary health care, health equity, and our extraordinary fireside host. Dr. Phil Felgner is director of the Center for Vaccine Research and Development at UCI And Dr. Michael Buchmeier is Professor of Medicine and Bioscience and the Associate Director for the Center of Virus Research. He has worked on coronavirus research for 40 years. We have started this in the past with a quick check-in with our speakers, reflections from the week. What are your thoughts? What have you learned? What do you want to share with us? And Cindy, I'll start with you.
Well, good evening, everyone. It's a beautiful day in the neighborhood, right? The sun was shining and it was really lovely for us to get dried out from all this rain. So I'll say from the primary care and health system perspective, we have been bracing and preparing intensively for what we anticipate may be a surge in the number of coronavirus patients with coronavirus that are coming into our health system and hospital. The good news is that the surge so far has not been as intense, as high, as great of numbers as many other locations have had. The challenging part is there's still so much that we don't know, that we don't understand. And there's a lot of unknown future for all of us in terms of what's going to happen. And I think that's one of the challenging pieces. Let me just say that the other piece of good news is that we feel, the health professionals and physicians, nurses, and others who are on the front lines, we feel so encouraged and supported and really appreciated by the community. It's just been heartwarming for us to see the outpouring of support in terms of making masks and delivering food and sending appreciation into the evening. So thank you for that. It helps us have the strength to go on and do the work that we must do to care for our patients and our community. So yeah, it's we're many weeks into this, and we know it's not over. There are many weeks ahead, and much to be learned.
Thanks, Cindy. Thank you so much. So, Michael, what can you tell us?
I am surprised, and I'm actually a little sad that the university as a community, with the students, with instruction in classrooms where you actually talk to people face to face, is not here. I'm hoping we'll get back to normal. I'm hoping that this isn't used as a justification to increase the amount of electronic teaching that we do, but we shall see. But it's not the same. It's very clear that the community is missing something right now. We don't. It's almost like you're caught in a time lag where you've lost track of the time, of the day of the week, of what I'm going to do today. And if you don't check your schedule every day, and I'm sure Eric is smiling because of that, you know that's a problem.
I know it's Friday when it's trash day, because it's trash day. That's about it.
Okay, thank you. Bill, what about you?
Yeah,
Week?
I'm losing track of the weekends. Everything's running together so fast. I get up in the morning, I'm answering emails even before I take my PJs off. I don't change it until about noon usually.
PMI, Phil, PMI.
Yeah. Well, it's been a very busy time. We've been never been so busy before, and but I've never felt so useful before. It seems like the work we're doing is important, and we're dedicated to it, and everybody is committed to help out as much as we can.
Thank you. Well, let's get to the questions then. General safety and contamination is on all of our minds. My first question is, when is it okay for us to rejoin our nearby family members? We're all feeling the loss of physical and tradition. Do we know when we can go back to sharing with our families?
I know that my family is not infected. My daughter is down in Encinitas with her family. They live in downtown LA, but she had the opportunity to take our other house and nest there. My son is up in LA and he's safe. So I wouldn't hesitate to meet with them tomorrow. But I think in terms of when we can do this in general, when we can restore normal activity, I would wait for the science to tell you and not take the political expedient. And we don't know that yet. I would be more comfortable if this went until the end of May rather than the 1st of April, 1st of May, as is being discussed apparently in the White House. So, you know, the commerce will come back. The economy will recover. You know, we recovered before from a recession, but the people that are sick and dying will not come back. And I think we need to be aware of that and really exercise that caution.
Profoundly said. Cindy, what do you think?
Well, I'm looking forward to the vaccine, so I'm looking forward to hearing from Philip. But I will say that to me, this has been really an extraordinary time because really our entire society has almost been put into suspended animation in lieu of trying to save people's lives and save people's lives who are most vulnerable to this infection. We know that we are not over the peak yet, and we know that this infection will probably continue to circulate until we have either reached a sufficient level of herd immunity, that is the majority of the population being immune to this virus, or we have an effective vaccine. And we know that both of those are far off into the future. I will say that our social distancing has worked because our hospitals are not overwhelmed. We have ventilators in capacity, and we're standing by ready to care for patients if we do indeed encounter a surge. But I think the fact that we haven't had such a rapid increase in numbers is really a testament to each and every one of you following the physical distancing recommendations. And thank you very much for that. You're saving lives.
Phil, did you want to add to that?
Well, with regard to vaccines, there's really not much we can do to shorten the timeline on vaccines. You know, it's going to take more than a year before we have even a vaccines in testing. So what our lab is doing, in addition to bringing our technology to development of one of the probably what is going to be many different vaccines, is to understand the herd, what you referred to as herd immunity. You know, what does it take to develop an immune response naturally that would confer protection to individual people and then to the population on the whole. And there's just so many questions, you know, that we don't have answers to. You know, we want to go out and test people to see whether they're exposed. So people, the market is being flooded with lateral flow tests now that will show either a positive or negative signal on there. And you're want to get some comfort from that to say that if it's positive, you're protected. If it's negative, you're not.
Can I stop you for a second? I want to continue in our discussion, the questions we have on general contamination and safety, because the diagnostic and treatment and vaccine aspects of this, there are some specific questions. So I'm going to just ask you if we can follow through that process. So there are, because there are so many people, and Cindy, you mentioned it, making masks and talking about masks and wondering about masks. And I think we talked about that, Mike, before the discussion opened earlier. What is an effective mask? So are commercial surgical masks reusable? Are scarves in lieu of masks effective? Are they reusable without washing them? I'll start there.
I think that the verdict on masks is that any mask will reduce the amount of spray, if you will, when you're talking, whether it's in the form of micro droplets or whether it's in the form of large droplets. A mask will catch a lot of that. And that's been proven over and over again. I think the old range estimates that we used to rely on of 3 feet for droplets and 6 feet for aerosol are probably an underestimate for aerosol. They're probably pretty close for droplets. You can see those easily. But the aerosol will float in the air for a long time. How about in the situation that you're in? You've got it, and we talked about this before. If you're walking down California and you need to get out and get some exercise, and you have a mask on or don't have a mask on, and you pass someone, it's very unlikely that that's going to be a significant contact. And the reason for that is dilution. Dilution and natural air currents will carry that away very quickly. So I think what we have to do is focus on close range. And close range would be if you're in the grocery store or you're in a congested area and you need to protect yourself and hopefully the people around you. If you're not infected, you're really protecting yourself. And so the question is the mask protecting the mask user, or is it protecting the people from the mask user? And I think that the mask is much more effective at stopping droplets than it is at really protecting the person wearing it. So if you breathe in, you're going to pull in whatever's in the air. If you breathe out and you cough and you sneeze, it's going to be caught in the mask. There will be some escape, there's no question about that, but there was a nice story in the LA Times this week that showed that the amount of escape is really fairly small, and it decreases very quickly with distance. So, you know, you do the best you can. No prevention is perfect. I see Cynthia shaking her head. What do you think? Cynthia, do you agree or not agree?
No, I would agree with that. And certainly from the medical side, we know that the N95, a mask that seals completely around your face, is most effective. And especially with the filter size, the micron size of what is filtered through what you're breathing. Obviously, we still need to be able to breathe, but the more we're sealed in with a very fine mesh, the less particles are able to enter that mask. We don't have enough N95 masks for the entire population, and it's not really comfortable to wear an N95 mask for very long. You kind of start to feel like you're suffocating in it and just want to take it off. So, as we've heard from Michael, any kind of filter is going to reduce droplets that are coming out. And I've been paying more attention to, little droplets just when I'm talking. I've been noticing, little droplets come out as part of our speech. It's a normal thing. It's not necessarily a bad thing, but it happens to be a very effective way of transmitting this virus. Right. And so anything we can do to reduce that droplet projection will reduce the transmission. And again, we're not entirely clear on the dose required to actually cause an infection. Probably the larger the load, the greater the likelihood of infection, but also our immune system and many other factors weigh into whether if we get exposed, do we actually become infected by an organism.
Thank you. Thank you. So protective equipment such as face shields are produced here by UCI as well as in large numbers by Apple. how do they compare to face masks? I have to say, I have a friend who's a critical care nurse at Kaiser, and she's been sending me pictures of herself in these face shields.
But she's also wearing masks. Wearing mask.
Okay. Yeah. What are your thoughts?
Well, I'll just say that normally we wear a shield if we're trying to protect our eyes from droplets, and usually a shield is worn along with a mask underneath it so that we can't have droplets coming from underneath. The shields themselves are effective in terms of preventing that droplet, but it's a matter of can things come around the edges of it, because the shield is not sealed in. So that's why you generally use a shield with a mask.
I see.
Because droplets even coming into the eyes, the conjunctiva, that can produce an infection. So we want to shield the eyes, the nose, and the mouth, all of our mucosal surfaces, otherwise known as the T-zone across the face, the eyes, nose, and mouth. Those are where the viruses can enter.
That's helpful. Mike, we were talking earlier about distance. There have been varying studies on safe social distancing. What is safe for inside versus outside versus athletic exertion?
I think athletic exertion, if you're on your own, if you're away from any other potential contaminating source, athletic exertion is good. You're exchanging air, you're getting rid of built up residue in your lungs. If you're not infected, you won't do any harm to your lungs by exercising, certainly. If you're indoors and you're in a crowd, it's probably a bad idea because of the potential of concentration. You're breathing much harder and you're breathing more deeply. And the deeper this virus gets into the lungs, the worse the pneumonia can be. It will get there eventually with many people, but if you start out that way, it's a much more severe infection. I think the other thing that I would say about the N95 is that when you buy an N95, you're supposed to be fitted by an occupational therapist or a public health official or someone who's trained to do that. And the risk that you run is actually that you will do some lung damage by trying to breathe through an N95. It is a very high resistance kind of respirator or shield. And so you do have to be careful with that.
Thank you. We have, I'm just checking the time, and we have quite a few questions more in the areas that I mentioned before. So let's get to them. I think a lot of these, Phil, you can help us with. Initially, what is the diagnostic process for COVID? And why didn't the CDC tests work?
Well, they're getting a lot of blame at the CDC for not having the tests prepared. They were prepared with coronavirus, but they had a strategy for detecting the breadth of the coronavirus infections, not just the SARS versions. So when they were confronted with this, they had a small number of tests. They only usually need a small number of tests to usually confirm that somebody does not have SARS infection. But they were unprepared. I know they take a lot of blame for this, but they corrected it. People say it's not that difficult to figure out what the right primers are for a virus like this, and that's true. I mean, there's a lot of really smart people who know how to develop these specific molecular tests, and they did it pretty quickly. But the pace that this infection is going, if you're wrong for just a matter of days, the consequences are really serious.
But Phil, now we don't have to send the samples to the CDC. It can be done locally, can't it?
Yeah.
Mhm.
I,
Was
I,
that everything?
I don't know how you feel about this, Mike, but they, I feel that the response is pretty fast now. What do you think?
Well, I think that now CDC has kind of gotten their act together. They still don't have enough tests to test everyone that's going to need to be tested, but there are enough privately administered tests that probably we can make up the slack. I think we're doing it here now, Cynthia, aren't we, at UCI MC?
We are, we're processing tests at UCI. A
Right.
number of different types of testing is
Yeah.
being done with different turnaround times in terms of being able to share results.
Yeah. I think the contrast between the American response, the US response, and the Korean response is what a lot of people point to. And I think the difference is that Korea paid attention to the information that was widely distributed, widely published, that said that these viruses are there and that the potential for another epidemic is really code red. And the last paper that was published about that was published just 2 years ago. And since then, we've had an epidemic of porcine pig coronavirus that killed 25,000 pigs. So that could have been interpreted by some as a warning because it was certainly on the radar.
Was it China, Mike?
No, it was porcine epidemic diarrhea, and it killed them. 25,000 pigs.
In China or Korea?
In China. But Korea is, believe me, is watching the pulse of Chinese disease to try to anticipate. Korea was able to give 140,000 tests in a very short period of time. And as far as I know, the results were reasonable, were reasonably accurate. But when you have a series of reports that are in the literature that report that there are up to 160 different coronavirus species that exist in bats in China, and that the live animal markets are still in operation, which they were supposed to be banned, then you've got a problem. You've got a potential problem. And that's the kind of thing that the PREDICT program, which was the program that was canceled in September 2019, should have picked up. And we're hearing today that there was a warning that came to the White House sometime before the new year, and it was ignored. So I don't know. Maybe this is from that data, maybe not. I would not give them credit for it unless you actually see a response that incorporates that information. But if the information is out there, it doesn't matter where it comes from, as long as it's there. And many of these papers were written by scientists in the U.S. and in Hong Kong. So it's not unusual.
Thank you. I appreciate that information. I want to go back to the individual questions and keep on the format. What is the individual health benefit to getting tested for the coronavirus in terms of treatment, other than the inhalers and nebulizers a person with a mild cough and or no fever could get without a positive test result?
So let me just say that because our testing capacity has been so limited, we know that the number of people who have COVID infection is probably much, much higher than what has been officially reported. We know that the majority of people who get the infection, again, thank goodness, the majority of people recover and they don't require hospitalization. But for the people who do become very severely ill, those are the ones who are most likely to have require ventilators and so forth. Why do we need to know whether a person is positive or not? There are a number of reasons for that. Number one is if you are a person of a high risk category and you know you're positive, you might be able to take more careful precautions in terms of monitoring and looking for symptoms of that would signal a person would be at a higher likelihood of really getting into serious trouble. The other reason for testing is for contacts, right? So if I've been in contact with someone who was positive, I would know that I need to be under more careful surveillance. And for example, as a family doctor, if I've been in contact with someone who's positive, I know and if I can get tested and find out I'm negative, it's a bit of a reassurance that I'm not going to be an asymptomatic transmitter to other people that I'm caring for, right? There are a number of challenges, though, even with the tests, is that what we call the sensitivity, that is the number of patients that actually have the disease that test positive, that's only about 70% for our tests. So our tests are not very highly sensitive. And so even having a negative test does not ensure that you don't have the infection. So we still have a lot of really serious problems with the testing in that, number one, there's a very limited availability of testing. And number two, the sensitivity of the test is not that great. And number three, the antibody test, we are not sure yet if having positive antibodies means you're immune to getting the infection, you've recovered from it, you can't pass it on to others. So there are a lot of unknown questions about the testing at this point, which makes it very challenging.
Phil, do you want to comment on that?
Yeah, I would just reinforce that comment that, you know, we can make predictions if we have antibodies, we'll be less prone to severe disease, or will have less a tendency to be transmitting the virus. But we don't know that for sure. You know, there's hypothesis that if you have antibodies, you might be even more prone to severe symptoms. You know, that kind of paradigm is common in dengue virus.
But Phil, is there any evidence that this is like the dengue virus, where that would actually happen? I mean, from other coronaviruses, is that?
Yeah, there are some publications that say that if you have pre-existing antibodies, you're more prone to severe disease.
Well, that's for
even those papers, you know, that it's going to take a lot more validation with our modern science to validate those things and find out how important they are. So a problem we're having is that we don't have enough information yet to make good decisions about how to manage this, both from a personal perspective as well as from a public health perspective. But the pace that everything's moving, I'm actually very optimistic. I mean, we're all pessimistic here because the circumstances are so terrible, you know, but this has only been going on for like a month, you know. So I don't know whether to be pessimistic because of the circumstances are so terrible and so many people are dying. I mean, certainly that we don't like that, but our ability to respond with the science that we have is also impressive. And you know, I think in a matter of a few months, we'll have so much more information about how to handle the circumstances well, both on a personal level, you know, so that we'll have tests in place that will actually help us have a better confidence that we're more resistant to the infection. And on a public health level, I think the herd immunity idea that we like to try and get 50% of the population above a threshold antibody level, if that antibody level is actually protective, you know, we're gonna have to know that. But the tools are in place for us to get those answers and get them really fast. So there's plenty of reasons to be pessimistic and depressed, but there's also, I think there's optimism that we're going to have the right answers and come up with good treatments and good ways of managing the infection.
Thank you. I got to tell you that the facilitator in me is going back and forth. I really want to hear all of you and all the knowledge and information and wisdom that you have to present. And I also want to honor the questions that have already been presented. So allow me to try and balance those two things in my own head and in front of you all. There are many questions to be asked. So Phil, to that, the next question is, can you tell us the latest about antibodies? And I think you spoke to that. Do mild cases produce as many as severe cases? Do the antibodies give us immunity? How long does the immunity last? If we think we've had a case, is there a place we can get tested for those antibodies?
Well, all those questions we don't have good answers to, but in record time, our hospital is getting approval for an IRB to probe everybody in the UCI MC hospital for their antibodies. And we're going to be collecting samples longitudinally from everybody too. So some of those people are going to be negative when we test them first time, and then later they'll become positive. When they're positive, we'll be able to check them for virus if we can actually still detect the virus in the mucosa. And that'll help us, because one hypothesis is that if you have antibodies, you won't have any virus. But we don't actually know the answer to that question. And so that'll be one thing. Well, in the hospital, we're expecting that there'll be a larger percentage of the hospital population that are seropositive to the virus compared to the average population outside. But for the sake of speed, we're getting approval for the hospital cohort, and then I think pretty soon after we'll start sampling the community and then we can compare what is the herd immunity when you're confined to the hospital setting, and what is it like outside in the community. I think another thing that's coming out of our research already is that the antibody response to severe cases is really very different from the response that's induced by exposure, asymptomatic exposure. So we really have to understand that. We have to understand what we know is a big difference between severely infected people and the mildly exposed people, and whether they're both protective, or whether one or the other is able to confer protection. So it sounds like a difficult problem, but it isn't going to take us that long to get those answers, especially at the pace that everybody is charging to get the answers, get those answers as soon as possible.
Well, and that is one of the questions is what is the timeline? What do you anticipate? When you say not that long, what do you anticipate?
I think some of these fundamental questions in 3 months, we're going to have pretty good answers to them about immunity, induced by exposure and a severe infection. We're not going to be done, because the things will come out. The papers are just flooding out. They have the bio archives paper. We published last week one, and then Monday we're going to publish another one, and probably the next week we're going to publish another one. And then they're not peer reviewed, you know, so we don't like to make a habit out of that. But this is a special situation we're in right now, you know, to get the information out there so other people can react to it and make a rapid response.
Yeah, but Phil, you're not just looking at what the responses are, that you have a response. You can actually identify which proteins in the virus
There are
people respond to.
Yeah, they're surprising things that come out of it. I don't know if it's surprising or not, but when you get granular detail, you can provide that information, you know, which if we didn't have the information, we couldn't, you know, speak to it. But you know, we've, I don't know, Mike, maybe, or you've run into this, and nuclear protein is a very sort of diagnostic antigen in these severe coronavirus cases.
So the next series of questions, sorry, Mike, I'm doing it again. The next series of questions are, and I do want to hear your answers. I want to come back to this. I just want to, again, honor the questions that have been asked. The next series of questions are around current drug trials and other and promising drug trials. Comestat, Aslate, ivermectin, hydrochloroquine, again, the president has been discussing.
Are you asking me? I'm sorry.
I'm asking both of you. I'm asking all three of you.
Okay, we've actually worked with hydrochloroquine, and we published on it back in 1997. And it was a very, very mild inhibitor of cell, of basically a movement of molecules within the cell. We were looking at endosomal uptake. And it was only marginally better than ammonium chloride, which is just a pure chemical response. They're anti-malarial drugs, and so they inhibit cell transport. The problem with them is that you have a very narrow window of therapeutic effectiveness. And as was reported in today's paper, people are reporting that some of the patients treated with hydrochloroquine develop heart murmurs and develop arrhythmias. And so we have that problem. And so, you know, there are reasons not to use chloroquine that are probably stronger than the reasons to use chloroquine. The other is, of course, that we use chloroquine in autoimmune diseases, systemic lupus, for example. And when we give something for an autoimmune disease, we're trying to suppress the immune response, which is not what you want to do in a serious viral pneumonia. So there are a number of reasons not to use chloroquine. The drug, the Ebola drug, is looking very good right now. And a colleague of mine down at University of North Carolina has been testing that in animals. That's Ralph Barrick. And it's looking very good. So if that one comes through, you can forget about chloroquine, because this is a specific adenine inhibitor that will basically cause the RNA to halt, to stop replicating. And so you can dose an individual with that because it doesn't affect the human. It's an RNA synthesis inhibitor from an RNA template.
Thank you. Thank you. What about the ivermectin or Comestat silate? Did you answer that and I missed it?
Cynthia, if I'm wrong on this, tell me, but ivermectin is another anti-arthritic drug. Mm-hmm. And why would you give an immunosuppressive drug to cure a disease that you need an immune response for? In the case of SARS, we learned in 2003 that about half of the cases of SARS in China were in physicians and other medical personnel, and many of those physicians were self-dosing themselves with steroids, and those steroids weakened their immune response to the point where many of them died. And that was the consequence of using the wrong drug in the wrong situation. So, you know, you may feel better because the immune response is dulled, but you won't clear the virus. And the important thing here is clearing that virus.
Thank you. So there's questions around the use of ventilators. Does UCIMC or either of the Hock hospitals do rank ordering of access to ventilators based on who's in the ER or ICU? Or is it possible to get the ER with extreme difficulty breathing and with extreme difficulty breathing and severe pneumonia symptoms, get tested quickly for coronavirus and jump the line over people with lesser symptoms, but who get tested far earlier in the progress of the disease, and who therefore entered the ER and de facto got in line for the ventilator treatment? It is my understanding that there's no lack of ventilators at this time. And thank you for bearing with my reading there.
So the bottom line is we have plenty of ventilators for the time being, for the foreseeable future. And because the peak of the curve has been reduced, the number of people who need ventilators is still much lower than the number of ventilators we have. So the great news is we've got plenty of ventilators to go around. There's no shortage for the moment. And California in general has what we expect to be, at least for the time foreseeable future, a surplus of ventilators. That's why we were able to share some with other states. Now, I will say that if we got to the point where there are more people requiring ventilators than what are available, then we have algorithms that help us, the physicians, determine who has the greatest likelihood of surviving. And we tend to treat those people that have the greater likelihood of surviving. And for people who we know are highly likely to die, whether they have a ventilator or not, those are the people who are less likely to be put on a ventilator if we get into that shortage situation. And we're not there yet, and we hope we don't get there.
That's true.
I have a question about the ventilators, actually, because I'm not involved in that type of thing, but I hear that when a person is put on a ventilator, they have a certain probability of survival probability. What is the mortality rate if a person gets placed on a ventilator in the hospital?
Well, again, that's highly variable depending on the person's underlying situation, right? And so I have a close colleague who's a physician in Seattle who reported to me that the mortality rate of patients who were placed on ventilators in Washington State was about 50%. So that means that if at least in that population, the patients who were placed on ventilators, about half of them would die. Now, there's a lot of variation in terms of the mortality rate of a ventilator based on your underlying lung situation and many, many other factors. It seems like I don't want to get into a lot of technical details for this group, but there's a lot of questions about whether the ventilators are as effective for this disease as some other types of pneumonia, because the SARS, the infection basically triggers this really inflammatory response in the lungs. So it's not so much the infection that's shutting the lungs down, it's the inflammation that comes from the infection that really damages the lungs' ability to exchange oxygen. And so that's where people get into serious trouble. And so, yeah, it's the mortality rate is quite high. And it's quite variable. And I think even that 50% rate has to be taken with a grain of salt, because a lot of the patients in Washington State were quite elderly, 80 years plus. And we know that the mortality for older people is much higher than for younger people. So if a 30-year-old gets put on a ventilator, their chances of survival are probably much higher than if an 80-year-old had to be placed on a ventilator.
Mhm.
Okay.
So the percentage of cases that actually end up on ventilators is fairly small too, isn't
It's
it?
very small. It's
Yeah.
very small. You know, out of you know, if 100 people get infected, 80 of them are going to get fine on their own. They don't need anything. They're just going to need to stay home and drink lots of fluids and take it easy, and their body will recover. And so for those 80 people that are going to get better on their own, it doesn't even matter whether they're tested or not, because we don't really have any effective treatment for the virus. So the testing for those people who have the milder symptoms doesn't really even matter. I mean, it's academically, it's of interest. You know, we'd like to know how many people are infected, but it's not going to make any difference in the outcome. It's for the 20% that have more severe infections. That's where we get into trouble with, you know, about half to a quarter of those will need a ventilator because their lungs just can't exchange oxygen efficiently.
Thank you, Cindy. Thank you. There were a number of questions that that you answered in that process. So I am going to go on to statistics and spread. How likely is it that the coronavirus might be spread asymptomatically through asymptomatic coronavirus positive individuals during the potentially 3-week long incubation period via aerosol-sized particles that can stay in the air for hours or longer, from which people can be protected with N95 masks? You answered those questions. You gave us some answers to that in a different way. I just wanted to circle back and make sure that we are clear about that.
Michael, do you want to take that one?
Oh, Cynthia, go ahead, Cynthia. I mean, where do you start on that one?
That's why I want you to answer that one.
Okay. First of all, those particles will stay in the air for a finite period of time. They will either disperse, or they'll dehydrate, or they will fall. And you know, they don't stay in the air for hours at a time. That's crazy. I'm sorry if I insulted anyone there. But the second thing, what was the second part of the question again? Susan?
A positive individual during the potentially 3-week long incubation period via aerosol-sized particles that can stay in the air for hours or longer and form which people can only be protected with an N95 mask. I have to tell you that I see this question as the potential for multiple conversations that may have come up, and it is the kind of question that worries people and keeps us thinking in a process there. So I think you have answered that in a variety of other ways.
Let me just
There's something else that
let Cynthia correct the length of the incubation period.
yeah, that's what I wanted to do. Yeah. The incubation period is not 3 weeks.
No.
We know that it's somewhere maybe 3 to 7 days, something like that, from exposure to acquisition of the infection. So it's not 3 weeks. So that's the good part. But we also know that some people who are infected are asymptomatic or have very minimal symptoms. So it may be hard to know if someone's been exposed and infected when the symptoms are very mild, particularly for children. We know children are great vectors, but they often have very minimal symptoms, like they have a bad cold, and they may be transmitting the virus.
And that's important. That's important to recognize that children, I think the last statistics I saw said that no one under 9 had died. this disease, and that the infection in many of these people is very, very mild. That doesn't mean that they're not symptomatic. That's the host immune response that's not severe. It's not the virus infection. The virus infection has not triggered the kind of a response that you get in the adult. And one of the things we need to study is why that difference occurs. You know, why a prior exposure may pre-sensitize you. That's the business of relationship with dengue. Why repeated exposure to related viruses may trigger a response. There's many, many reasons for that. And I like to, let me just say one more thing, Susan.
Okay.
Studying an epidemic like this is like studying a plane wreck. You know, the crash is accompanied by a lot of damage, a lot of angst, a lot of noise, a lot of activity, but it isn't for 2 to 3 years before you figure out what really happened. And that's what's going to happen in the next 2 to 3 years.
Yeah.
And that's why antibody is important to study. That's why immune responses are, drugs, vaccines. We had vaccines that worked in primates from the SARS epidemic, but they were never adopted because the epidemic disappeared and there was no market for the vaccine. Novartis had a wonderful vaccine, and they published the data. They put it in the context of a meeting that was held in October 2007, presented by a man named Unger. And he's now working for GlaxoSmithKline. He was with Novartis Vaccines then. And it was a stabilized glycoprotein subunit that could be produced with no cytoplasmic tail, and that generated a useful immune response. This isn't rocket science.
So, good question. We are at 2 minutes, and again, Cindy has taught us to honor our 1 hour and
get a good night's sleep.
He had to jump in. I was just asking Michele if he was able to save our chat. I think there are a number of good questions in there. I will still go back through our questions and mine them for the ones that did not get answered. And Cindy, Mike, and Phil, if you are willing to pick this up next week at this time, same bat channel.
Sure.
Then we can do that. Right.
Where else are you going? Really?
Yeah, right.
Yeah,
I just have to keep track of which day it is.
You know, we're all feeling that, Phil.
Flash goes out on Fridays.
Excellent point. Thank you. This has been wonderful, and I really appreciate, despite my facilitation, I value the science very much. I really do, because I
don't
got
give
this
her a
guy.
choice. That's why.
So at this point, I will say good night to you. Are there one last thing that you would like to say to us, Cindy, Phil, Mike?
I think be patient. Follow good advice. Good advice is almost inherently not political, and use your best judgment. And remember, dilution is the solution in terms of breathing.
That's brilliant, Mike.
Okay.
Cindy, Phil?
Bill?
Well, keep a lookout for the science. There's going to be a lot of good answers coming along here.
Thank you.
Thank you. Cindy?
And I'll just remind us that physical distancing is not the same as social isolation. So it takes extra effort to reach out and connect with your neighbors, your friends, your loved ones. And those connections are really important. They boost our immune system. So take care of yourselves, reach out to your loved ones, and thank you for everything the community is doing to flatten this curve.
Thank you, Cindy.
Good night.
Hobart, is there anything you wanted to say? One last thing? Take us out?
I'm sorry?
I asked Hobart if he would like to jump in, take us out. Oh,
the Zoom bar. Somebody.
Tea. It's tea.
Okay. Now, just thank you, everybody. It's been really wonderful. and to donate your time, your effort, your knowledge. It's been a tremendous way in which we can pull together as a community. Thank you so much.
Thank you. Good night, everyone.
Good
Good
night.
night. Good night. Cheers. Cheers, Michelle.
Thank you.
Bye. Bye.
Cheers, Michelle.